Plan for today is to finish, hopefully anyway, our lecture on biochemistry, and if possible, start our last lecture of the unit, cells. If not, we'll get to it on Friday. So, on Monday, we got to talk about some of our inorganic compounds and features of them. Remember, inorganic compounds are one of the two categories, the other being organics. We'll talk about organics after we get through acid, bases, and salts. Water was our, most important inorganic compound as far as, with the exception of oxygen, but water is super important, not only in us, but all living things. One thing that we talked about that water does is it dissolves certain things, okay? Things like acids, bases, and salts dissolve in water, and that's because they dissociate, okay? Which is a fancy word to describe the fact that those bonds, like sodium and chloride and salt, those those ionic bonds split apart, okay? And so what ends up happening are the ions become separated and surrounded by water molecules. Okay, an acid is a substance that when it dissociates into one or more hydrogen ions, which is abbreviated H+, and one or more anions. A lot of times acids are referred to as proton donors because a hydrogen ion is simply just a proton. We think back to when we talked about hydrogen, and I introduced it a little bit in our chemistry lecture, we learned that its atomic number was 1, right? Atomic number 1 tells you it has 1 proton, also 1 electron. Okay, but now it's H+, so that means that there's no longer equal numbers of protons and electrons. Okay, what's taken place is it's lost that electron, which is not a bad thing. It gets rid of it to make it happy. But hydrogen typically doesn't have a neutron. So what's left over is just that 1 proton. That's all that's left. So that's why a lot of times you'll just see hydrogen ions listed as a proton. And that's why they're called proton donors. Acids are, that is. How acidic a solution is, or the level of acidity, is dependent upon the number of hydrogen ions a particular acid releases compared to what's called hydroxide ions, OH, that you can see has a minus sign associated with it. So I've given 2 examples to demonstrate what can take place, and we'll decide whether or not we're dealing with an acid. H2O, we all know, is water, right? Okay. If you dissociate water, we form 1 hydrogen ion and 1 hydroxide. Okay. Well, if you look at the difference between the 2, or you look at the ratio, there's 1 hydrogen ion, 1 hydroxide ion. Okay. To be an acid, we need more hydrogen ions than hydroxides. 1 and 1 are equal, correct? So is water an acid? No. Good. Okay. HCl dissociates to form hydrogen ion and chloride ion, where there's 1 hydrogen ion and no hydroxide. So it's a 1-to-0 ratio. So there's more hydrogen ions than hydroxides because there's no hydroxides. So HCl is a acid. I told you the other day it's hydrochloric acid. Okay. If we're dealing with a base, which is the opposite of an acid, this time we dissociate the ions, and what's taking place is the ratios in favor of the hydroxide, OH minus. Okay. Another term for a base is an alkaline. So when they dissociate, they form 1 or more hydroxide ions and 1 or more cations. Instead of calling them proton donors, we call them proton acceptors because they remove hydrogen ions from a solution. Sodium hydroxide is our example at the bottom. It dissociates to form a sodium and a hydroxide ion. 1 hydroxide, but there's 0 hydrogen ions, so it is in favor of hydroxide, our ratio, which means sodium hydroxide is in fact a base. Now going back to water, is that a base? Are there more hydroxides than hydrogen ions? No. So water is neither an acid or a base. What is water? It's what we call neutral. It would be in the middle of what we call our pH scale, the scale that tells us whether or not we're dealing with an acid or a base. The term pH, notice it's a lowercase p and a capital H, is an abbreviation for what we call power of hydrogen. It's used to mean the relative hydrogen ion concentration of a solution. It's talking about that ratio. If there's more hydrogen ions than hydroxides or vice versa, or if they're equal. Okay. It's a very complicated logarithmic scale that you would have to use a calculator to calculate up. Great thing is, simply what you'll need to know it as is a 0 to 14 scale. 0 and 14, between them is 7.0, right? That's smack dab in the middle, which happens to be, as you can see, where pure water is. Okay, and you can see under the word neutral, it says hydrogen ions and hydroxide ions are equal. Anything above 7.0 is a base, which happens to be color blue in this picture. The farther you get from 7, the stronger the base is. So 7.1 is a very weak base. 13.9 would be a very strong base. Okay. Notice up, well, never mind, I wanted to say that. I'll just not confuse you. Below 7.0 are the acids. And the farther you get from 7, the stronger the acid becomes. So you can see lemon juice, gastric juice that are listed there in the figure, really strong acid, somewhere around 2, whereas urine, 6-ish. A fairly weak acid. Okay. As far as the table goes, you can see I've listed off, or the table has listed off, I didn't do it, pH values of selected substances. Some of these we'll get to know, like gastric juice, that's our stomach, very acidic, and that's because we produce the hydrochloric acid in there. You can see it can be as low as 1.2. I'm not going to ask you to know any of those pH values other than one, and that's blood, which has a pH of 7.4-ish, because as we know, we don't ever have a perfect balance of anything. Controlled conditions are never perfectly in balance. There's a dynamic equilibrium. Blood is 7.4, which makes blood what? A base, an acid, or neutral? It's not neutral. It's a base. 7.0 is neutral. 7.4 is not 7.0. Okay, it's a little bit above 7.0. So it's a really, really weak base, but still it's a base. Okay. There will be questions on the exam. I might give you a pH value of 4. You'd have to tell me, is that an acid, a base, something like that. You know what I mean? One of the most important controlled conditions in our body is to maintain pH of various body fluids, especially our blood, as I've mentioned, around 7.4. You compare the pH of your blood to the pH of your mouth, to the pH inside your stomach, very, very different. And they all have their own very narrow limits in which they have to be maintained at. Okay? We like to keep our blood right within 0.05 above or below. So between 7.35 and 7.45. So smack dab in the middle is 7.4, where it's perfectly where we want it. Okay. Any drop in blood pH below 7.35 is known as an acidosis, which sounds correct, but it's actually kind of misleading because 7.3 is still a base, right? Okay, but we're trending towards acidity, maybe because acids are building up in our body. Okay. Any elevation in blood pH above 7.45 is called an alkalosis. Okay. And our pH is constantly being disrupted Something as simple as holding your breath will change your pH. Okay, because if you do not breathe out the CO2 that you're producing, which you're constantly going to be producing, CO2 will build up in our body and combine with water and become an acid. Okay, so just by breathing, we help maintain pH, which is cool. Even though strong acids and bases are continually taken into and formed in the body, pH of fluids inside and outside of cells remains almost constant. And that's because we have what are called buffer systems. Okay. The function of them is to convert strong acids or bases into weak acids or bases. So basically, a buffer just helps maintain pH. Okay. And the great thing is, if you take a base and you add an acid to it, they neutralize each other, or vice versa. So they can convert strong acids or bases, and that's what buffers are. They work by removing or adding protons, which again, protons are hydrogen ions. Carbonic acid bicarbonate buffer system is one of the more important buffer systems, acts to buffer pH changes in our blood. We'll talk about pH balance and maintaining acid-base balance later on in the semester, way later in the semester, when we talk about the two major systems that do so. Anybody have a guess? So we learned about the 11, or if you call them 12, systems of the body. There are two that are most responsible for maintaining pH. Anybody have any guesses as to what they are? Come on, somebody guess. Very good. The urinary system is the most important for long-term acid-base balance. We can pee out acids in our urine. What's the other one? Nope. Not the nervous. I told you earlier we can breathe to blow off CO2, right? That's a function of your respiratory system. That's how we can maintain pH second to second, because we're constantly breathing off that CO2. So respiratory and urinary, sometimes kind of surprising to students. We won't talk about those two systems until way later in the semester. Salts, similar to acids and bases, but when they dissociate, like over here on the far right of our page, potassium chloride, the two ions that are formed here are neither hydrogen ions or hydroxide ions. And what's cool about acids and bases, when they combine, they can react and form salts. And salts are important in our human body to act as electrolytes, important for carrying electrical currents, especially in our nervous and muscular tissues. In our next unit, when we first introduce a system, the nervous system, we're going to be talking a lot about action potentials. The nerve signals, same thing. So we'll get really familiar with sodium, potassium, especially Na+, K+, and to a lesser degree, but still important, Cl- chloride. All right, that's all I'm going to introduce to you guys on the inorganic compounds. We'll move on to the organics now. The other principal class of compounds. The term organic should trigger just immediately from here on out thoughts of carbon. I'm sure you've heard of classes called organic chemistry. I think we teach any here or any of that. That's usually a pretty upper level class in chemistry. You're going to basically study carbon that entire semester if you take organic chemistry. And that's because organic compounds have a carbon backbone. I'm not talking about one carbon, talking about, in many cases, a whole bunch of them. Okay, and a lot of times you'll see hydrogen bonded to that carbon, forming what's called a hydrocarbon. Okay, down at the bottom, we have 4 different kind of boxes with pictures in there. You see carbons and hydrogens all over the place. Okay. Carbon is unique because it, I'll show you a picture here in a couple of slides, has that atomic number of 6. which means that it's got 2 electrons in its first shell, which is not really important, but 4 in the outside shell. And in that state, carbon is not satisfied, is it? How many does it want to have total? 8. And so carbon will seek out 4 more electrons. And it will do so in a variety of ways. Okay, it can bond with 4 single covalent bonds. It can form 2 doubles. It can form a triple and a single, a double and 2 singles. It can form chains and rings, and we can see branches. As you can see, the variety is pretty well endless, and we'll find out as we really get into this lecture. Okay. Now, think of organic compounds like a brick house. Okay, a brick house is made up of bricks, correct? Okay, each brick is what a monomer is. Okay, mono is a prefix that means 1. You take 1 and you bond it to another, you've got a dimer, right? We talked about this and we showed this picture on Monday. Okay, performing what's called dehydration synthesis is how we build these brick houses together. Take 1 and another, 2 bricks, makes a dimer. That's what we do to build them up. We can actually break them down by that hydrolysis reaction we talked about last time, like was shown there in the second figure. And so formed by dehydration synthesis that we talked about last time, dissolved by hydrolysis that we also talked about last time. Now, aside from just having carbon and hydrogen to help separate and make each of these organic compounds unique are what we call functional groups, which are special groupings of specific atoms that confer chemical properties to the organic molecule. Table 2.5 here in your textbook, from your textbook I should say, lists off some of the most important major functional groups in our organic molecules. Not something I'm going to ask you to know, but some of these we will get to know very well. Like, for example, phosphate, which is down there towards the bottom. Okay, that's what a phosphate looks like. You've got P surrounded by 4 oxygens and an R over there on the left. Okay, we have 3 of these in each of our ATPs because ATP is short for adenosine triphosphate, and tri means 3. Okay. We're going to talk about amino acids. Guess what's in an amino acid? An amino group, okay, which is shown down there at the very bottom. We'll talk about them, not in great detail, but I wanted to kind of just discuss what they are. They give special properties to the various organic compounds. Already kind of talked about carbon. It's the most versatile building block of molecules, and that's because it has that atomic number 6, therefore having 4 valence electrons. And so it can share up to 4 times covalently, making large complex molecules possible, with carbon being a central point from which the molecule can branch off into 4 different directions. It gives carbon covalent compatibility with many different elements. And so covalent bonds that link carbon atoms together in long chains form the skeletal framework for our organic molecules. And that carbon skeleton can vary in things like length, shape, like straight chain, branched, rings, number and location of double bonds, other things like those functional groups that we just talked about bonded to available sites. So this variation in carbon skeletons contributes to the complexity and diversity of our organic molecules. So let's get into them. First thing, I just want to introduce the 4 main groups, and then we'll go through them individually, one by one, beginning with carbohydrates, then into lipids, and then the proteins, and then finally nucleic acids. Okay, what are lipids best known as? Fats. And which are not necessarily a bad thing, despite what some people think. Over on the right side of our figures is an example of a nucleic acid. I imagine you guys can pick up what that is. DNA, you couldn't already, that's what it is. RNA is also a nucleic acid. We'll talk about those two things. And then ATP is it's kind of a hybrid nucleic acid. I'll explain what that means. Well, that'll be towards the very end of the lecture. Let's talk carbs. Carbs, unfortunately, have also become a hot button. People think that they're the total enemy. Don't get them in your diet, that kind of stuff. Carbs are good. As it states there in that first sentence, they provide most of the energy needed for life. Includes what we call sugars, starches, glycogen, and cellulose. We'll talk about each of those here in the next couple slides. Typically, when we talk about energy, we're going to talk about ATP, and that's what we use carbohydrates for, but some can be used to be converted to other substances that are used to build structures. Okay. Other carbs can function as food reserves. This is why athletes will carb load before an endurance event. Unfortunately, if we end up with too much carbohydrates and we eat a lot of sugars and stuff like that, we'll end up converting inside of our body those carbs to fats and store it as fat. And we have an unlimited ability to store fats. Some people take too much advantage of that. Carbohydrates are divided into three classes based on the length of their carbon chains. You got mono, di, and polysaccharides. Once again, mono means one, di means two, and poly means many. Let's talk monos and dis here on this page. Monosaccharides are described as the building blocks or the monomers of our carbohydrates. So these are the individual bricks for our brick house. Okay. They're typically pretty small and short as far as carbons go, 3 to 7, somewhere in that neighborhood. Glucose is a 6-carbon sugar, C6H12O6. You take two of those like glucose and fructose and you bond them together by way of dehydration synthesis. You got two now, you've got a disaccharide like sucrose, which is a fancy way to describe table sugar. Okay. Notice I've used the word sugar a couple of times on this page. Collectively, monos and disaccharides are known as simple sugars. Okay. They are found in sweet things like the Dr. Pepper that I drink on a way too often basis. Cookies and cakes that taste really great, but we should not be using them as the major form of our diet as far as carbohydrates go. What we should strive to get most in our diet are the polysaccharides, specifically starch. These are the largest carbohydrates, and we're not just typically talking 3, 4, 5. We can be talking about hundreds of those bricks. Like most houses that are a brick house are going to be, hundreds upon hundreds of bricks. Like glycogen, which is pictured here on the next page. Each of these little purple rings is a glucose molecule. So I'm not going to go through and count it up, but there's a lot in there. Glycogen is our principal polysaccharide that we store, and really shouldn't say liver or skeletal muscle because it's not one or the other, but it's both. So really and should be in there instead of or. Okay, this is what we do when we get lots of glucose in our body. We take it in and we store it in those two places as like filling up your gas tank in your car. That way, when you need that energy, you can call upon it pretty much immediately, okay? We break it down by way of hydrolysis to form glucose molecules. We break those glucose molecules apart, which then forms the ATP. And what's nice about storing it in our muscle is like when we exercise, muscles are where the ATP is needed. The glycogen is right there to provide the glucose to our mitochondria where we can then make the ATP. We'll get into all that stuff later on the semester when we talk about cellular respiration. Fun stuff that everybody loves to talk about. Other polysaccharides include starch, which is how glucose is stored in plants. Starch is found in things like pastas and potatoes, okay? Very filling, okay? Major carbs in our diets. Cellulose is a structural polysaccharide in the cell wall of plants. It's indigestible by humans, but does aid in the movement of feces to the intestines. And a lot of times, this is referred to as like fiber, okay? Now, since it's indigestible to us, does that mean it's bad? No, fiber is a great thing. Okay, here in Kansas, a great example of, and we're just a few weeks out from that. We did it a couple weeks ago, harvesting sweet corn. Everybody loves sweet corn typically. Okay, it's a kind of gross visual to think about, but corn comes in and it goes out the same way. And we don't break it down. We can't absorb anything really from it. And that's actually beneficial for us. Okay, fruits and veggies are loaded with fiber, obviously like fiber 1 bars as well. And they speed things up. And if you're not used to eating fiber, be careful. If you eat a whole bunch, you might be running for the bathroom. And it speeds things up, which is good because it reduces risks for like colon cancer and some other things like cardiovascular disease and whatnot. Chitin is our final, and really I won't get into it other than just listing off that it's a polysaccharide. It forms a cell wall of the fungi. It also is what gives insects their exoskeleton. Table 2.6 here on the page lists off those monos, dis, and polys and gives you examples of them where they're at. Some of them we did talk about, some of them we didn't. I won't get into it. It's not something I'm asking you to know. All right, let's move on to lipids, or fats, as they're also known as. Lipids are described as water-insoluble organic biomolecules, are generally nonpolar, making them hydrophobic. So there's kind of 3 major terms in that first sentence. I'll start with water-insoluble. Okay, I think we talked about this the other day, but if you pour water into the same container or you pour oil on top of water, what happens? The oil floats, right? Okay, that's because oil is a form of fat that is water insoluble. So what does water insoluble mean? Won't dissolve in this case, water. Things that are water soluble will be like salt, because water soluble means they do dissolve in water. Water insoluble means they don't dissolve in water. And that's because fats are typically nonpolar. We used the terms polar and nonpolar the other day when we were talking about covalent bonds. Remember, polar covalent bonds are where the electrons were not shared equally, whereas nonpolar, they shared them equally. And I told you guys, in this class, you can kind of think nonpolar and polar like no charge and charge. Nonpolar, no charge, polar means charged. The term hydrophobic refers to the fact that fats are, and this is how it literally translates, water-fearing. Hydro means water, phobic, think phobia, like it's afraid of. And fats and water don't get along, like cats and dogs, as they say. And that's all because they're nonpolar. So make sure you know what that means. This is something that we're going to get into a lot when we talk about the plasma membrane of our cells. Lipids typically make up 18 to 25% of the body mass in lean adults. Obviously, the key phrase there is lean adults. Unfortunately, many Americans are not lean, and so their body mass is made up of more fats. Fats themselves, or lipids, are composed of hydrogen, or excuse me, carbon, hydrogen, oxygen, but there's very little oxygen. And as I said, lipids are not necessarily a bad thing. They are good for us. They serve many functions. They store energy. 1 gram of fat stores twice as much energy as a gram of polysaccharide. We'll get into that here when I get into triglycerides. They're a more compact fuel reservoir than carbohydrates, which is why we can convert carbs to fats if we're getting too many carbs in our diet. Cushions vital organs like our kidneys, which are surrounded by a fat capsule. In our skin, there's fat to help insulate against heat loss. Not nearly as important as like, in whales that have blubber, but we've got a little bit of fat to protect us from the heat or from the cold. Aids in the formation of cell membranes, formation of hormones, and even aids us in digestion, which if you've ever heard of bile is something that is, can help digest fats. All right, let's talk about the different types of lipids, beginning with the simplest, fatty acids. Are here on this page. Okay, so thinking back to what we just talked about with carbohydrates, we said carbohydrates are built with building blocks called monosaccharides like glucose, which we take glucose and we build it up, we can make like glycogen, right? Okay. This section on fats is different from the other 3 organic compounds in that there is no universal building block that is used to make all fats. Meaning that we can't just take a building block and make any of these. But we can take a fatty acid, and actually it's multiple, and we can make these 2. So fatty acids are as close to a building block as we get when it comes to fats. And that's what is this sentence about. This is what this sentence is about. They're used to synthesize triglycerides and phospholipids. 2 forms of fatty acids exist. Saturated, which is what the palmitic acid figure is showing you. We'll look at all the carbons and hydrogens bonded together, carbons running down in a straight chain, hydrogens on the outside. Key features of saturated fatty acids is there's not going to be any double bonds located in that carbon to carbon to carbon to carbon to carbon. There's only one dash between a carbon to another carbon. That means there's only a single covalent bond. Whereas oleic acid right below it, notice where the acid or the fatty acid bends, there's a double bond. It kinks, if you will, because of the presence of at least one double bond. And that makes it an unsaturated fatty acid. Now, if you have taken like a nutrition class or you pay attention to what you eat, on food labels, a lot of times you'll see saturated fats, unsaturated fats. Okay. One of these 2 is really well known for being a lot worse for us. Anyone know which one it is? Saturated. Okay. So we try to avoid getting lots and lots of saturated fats in our diet. At least we should be. And that really brings us to this type of fat, what we call triglycerides. Triglycerides are the most plentiful lipids. So when somebody's talking about a fat in their body or a fat in their diet, most likely they're talking about triglycerides. And they are made up of 2 building blocks, glycerol and the 3 fatty acids, as shown here on this page. I'll bounce back and forth. So if you want to write out the word here twice, you can. This illustration shows what a triglyceride looks like. Here in our blue box, there's a 3-carbon molecule. You can see the 1, 2, 3 carbons. That's a glycerol. Okay. Attached to each of those carbons, it's not actually directly because there's an oxygen in between, but is a fatty acid. We call these tails or chains, okay, and there are 3 of them. Now you can probably figure out why we call them triglycerides, because tri means 3. Okay. If I go back to this previous page, glycerol molecules, 3 carbon molecule forms the backbone of triglyceride. The 3 fatty acids are attached by dehydration synthesis to each carbon molecule of the glycerol backbone. This bond is known as an ester linkage. Not something we would really need to worry about. But we can find triglycerides at room temperature, which, you know, right now this is room temperature, about 70 degrees, as either in a liquid form or a solid form. If it is a solid, we call it a fat. If it's a liquid, it's an oil. Okay. You guys ever, cooked or been around Crisco or lard? You don't see many recipes calling for those types of things anymore. This is what, like, my grandma used to cook with. They're really unhealthy. Okay, like, things taste great, not good for us. Okay, they are solid at room temperature, and that's because all of those fatty acids are like this, straight across, straight chain, because they're saturated. Oils like canola oil and olive oil and safflower oil are liquid at room temperature because they cannot solidify, and that's because they've got that little bend. They're unsaturated, or they have unsaturated fatty acids. Make sense? Okay. So if you do cook with lard or Crisco, throw it away. Go get some oil. Okay, as I told you just a second ago, they are the primary type of fat we have. They're the most highly concentrated form of chemical energy that our body has. They provide more than twice as much energy per gram as do carbs and proteins. This is why they are high in calories. Now everybody's heard of a calorie before, or what calories are. I could ask you right now, say, well, tell me what a calorie is, what that means. People generally don't know what to answer. Okay. Calories in food and drinks just tell you how much energy you can get from that substance. Yeah. The more calories, the more energy it could provide us. Okay. If you have a equal sized amount of carbohydrates and an equal sized amount of fats are going to provide more energy. Okay. What that means is per gram. So if you have 1 gram of carbohydrate, same thing for proteins, they can provide us with 4 calories of energy. Okay. Whereas a gram of fat, it's the same amount, it gives you 9 calories of energy. So that's actually more than double. There is a 4th thing that people can get energy from, which a lot of college students love to take advantage of, unfortunately can contribute to the freshman 15. What am I talking about? It's not a food. It's a drink. Alcohol provides 7 calories per gram. So it's not as energy rich, but it's close to fats anyway. And as I told you earlier, humans have an unlimited capacity to store triglycerides in our adipose tissue. So excess dietary carbs, proteins, fats, and oils will be stored in that form in our adipose tissue. Okay, our next type of fat, similar looking, you can see the picture on the right side of the page, to a triglyceride, but at the same time a little bit different. There are similar components that make up phospholipids. There's a glycerol backbone. If you look, I'll show where the glycerol is in the picture. This section right here, we can see the 3 carbons. That's what triglycerides had. Attached to 2 of the carbons are fatty acids. We had 3 fatty acids in a triglyceride. We only have 2 here. So the 3rd carbon is attached to a phosphate group and then some other stuff, which creates kind of 2 distinct regions to each phospholipid. What we call polar head, which is charged, because phosphates actually have 3 negative charges. And then the tails, which are nonpolar, just like any other fat. But the polar head is unique because other fats are not polar. Okay. Which is really important when it comes to exposing these things to water. And as we know, our body is full of that substance, water. The polar heads actually like water. They are described as hydrophilic, which I always tell students, just think smiley face water. They like it. They're attracted to water. Whereas the tails are hydrophobic, which we said are water-fearing, so I always say frowny face water. Okay. They don't like water, they're afraid of it. Okay. Just like any other fat. Now, why is this important? Well, they create and are the primary structures of our plasma or cell membranes, which is the structure that surrounds all of our cells. And they create a double layer of phospholipids, what we call a bilayer, where the tails are pointing in the same direction as one another, and the heads are pointing outward from one another. The heads point into the cell, which is full of water, and outside of the cell, which is where you also have water. This is very important for what we'll get into in our next lecture. The fact that phospholipids have both polar and nonpolar regions creates or makes them amphipathic, which is a fancy word. We'll get into these guys more later on in this unit. Let's talk steroids, which, looking at the figure at the bottom, or figures, look nothing like those first three fats we've talked about. Steroids are derivatives of a four-ringed hydrocarbon skeleton. That's what the ABCD here in each of those pictures is, okay, which makes them structurally different from the other lipids. We don't have chains so much as rings here. Okay. Most common steroid in animal cells, and yes, we're an animal, is cholesterol, which is the starting point for synthesizing all the rest of our steroids. Okay. Examples of steroids in our body include estrogens, which is a plural form of estrogen. Estrogen is typically what people say, but actually females have more than one estrogen, like estradiol, that's shown there. Testosterone, which is a very important male sex hormone. Cortisol, which is our stress hormone. Bile salts, which are a component of bile that helps us digest certain things. And vitamin D, where do we get our vitamin D? Aside from our diet, the sun. Okay. So that's the gist of steroids. We'll get into those more later on in the semester, like when we talk about hormones, like testosterone and such. All right, let's talk eicosanoids. Lipids composed of a 20-carbon unsaturated fatty acid called arachidonic acid. Three principal subclasses of eicosanoids exist: prostaglandins, thromboxanes, and leukotrienes. Prostaglandins play a role in regulating the effects of several hormones. Thromboxanes, like thromboxane A2, is the only one that we really talk about in this class. Are part of a process that we do when we bleed. They constrict our blood vessels and help activate platelets. Leukotrienes participate in allergic and inflammatory responses. So we really don't talk much about these other than just kind of a few times when, like we talk about bleeding and allergies and inflammation. So we'll kind of wait and learn about those later on in the semester. Other fats include fat-soluble vitamins, such as beta-carotenes, the yellow-orange pigments that you find in like egg yolks and carrots and tomatoes that we convert to vitamin A. Vitamins D, E, and K. So A, D, E, and K are the fat-soluble vitamins. And then lipoproteins. We don't really get into vitamins too much in this class. We don't really talk much about lipoproteins either. But lipoproteins are molecules formed when lipids join with a hydrophilic protein molecule. So it's exactly what it sounds like, lipid protein kind of hybrid. They're more soluble in our blood plasma because the proteins are on the outside while the lipids are on the inside. If you've taken nutrition, I imagine you've heard of HDLs and LDLs. You may have ever heard those terms before. And the L in the HDL, LDL, the last L, I guess, for LDL, is short for lipoproteins. And if you ever have your blood samples taken to like measure cholesterol and fats and that kind of stuff, they'll also check HDLs and LDLs because we need certain numbers of those in our blood as well. Table here on this next page just summarizes all those different fats we've talked about. So we'll move on from that. To our third organic compound class, proteins. Large, very complex molecules make up about 12 to 18% of a normal, and key phrase here is lean adults. Lean adult body, that is, instrumental in almost everything cells do. Okay, that's a key phrase I like a lot. If something's happening in the body, it's a good chance a protein's doing it. Okay, the building blocks to form proteins are called amino acids. There's an illustration there of an amino acid. You take one amino acid and you bond it to another by what's called a peptide bond. Okay, proteins are much more complex in structure than carbs or lipids. And there are 20 different amino acids. Each consists of 4 major parts. An amino group, the NH2 shown here on the left in blue. Carboxyl group, that's the acid, so there's where the amino acid term comes from. There's a side group or side chain, R group, whatever you want to call it. It's not like R is an atom, but it's more like a group of atoms. They just abbreviate it with this R box. And then there's a hydrogen atom, all bonded to a centralized carbon. Okay. Now, where a amino acid differs from another one is this R group. Okay. There are 20 different amino acids. You take 2 of those, you can make a dipeptide. A third makes a tripeptide. 4 to 9, that's a peptide. 10 plus are polypeptides. Okay. Think of an amino acid like a letter in the English alphabet. How many letters are in the English alphabet? 26. Okay, there's 20 amino acids, so there's not quite as many letters. Okay, here's a question you're not going to be able to answer. How many words are in the English language? It's an impossible question, right? How do you make words? Letters, right? You put them together in a specific sequence and number. Okay. Same thing occurs with making proteins. You take those 20 different amino acids, leucine, isoleucine, methionine, so on and so on and so on, and you put them together in a specific number in a specific manner, making a great variety of proteins possible, just like a great variety of words is possible. Okay. Each of those proteins that we make is unique in function, just like each word is a unique word, means something different, right? Okay. Chances are, and this is a phrase I really love, I already said it, if something's getting done in our body, it's being performed by a protein, which is demonstrated here with table 2.8. Table 2.8 is a rare time where I tell you to know a table. These are the 6 different types of proteins with their functions listed and examples of them. In anatomy, I'm getting ready to talk about keratin, a protein that's found in our fingernails, it's in our hair, it's in our skin. It's a structural protein that forms a framework of various body parts. Other examples would be like collagen, that's in our skin and it's in our bones. Proteins can be regulatory, okay, like the hormone insulin. I imagine everybody's heard of insulin before. That helps regulate our blood sugar. Insulin is a protein. Okay. Actin and myosin are two proteins in our muscles that contract and allow our muscles to shorten. So proteins can be contractile. Antibodies are proteins that circulate in our blood to help us fight off viruses and bacteria and that kind of stuff, so they can be immunological. Transportation. We'll talk about how proteins that are in our plasma membrane can bring stuff in and out. Hemoglobin is a transport protein in our blood that carries oxygen. And then something we're going to talk more about on Friday are how proteins can act as catalytic enzymes. In other words, they speed up chemical reactions. So we'll pick up with protein structure on Friday. Got a couple more slides on proteins, and then we'll move on to nucleic acids.